Hi everyone, welcome to my channel CAPSA. Today's topic is type of receptors. As a part of pharmacodynamics, we will understand everything about different types. So receptors are actually of four types. Okay, like first one is ligand-gated ion channels. Ligands are involved. Enzyme-linked receptor, where specific enzymes are involved. G protein coupled receptor, G protein is involved, and nuclear receptor, where DNA or transcription activation or inhibition is involved. Okay, so first one is ligand gated ion channel, second one is your enzyme linked receptor, and the third one is G protein coupled receptor or G protein linked receptor. And the fourth one is that is your nuclear receptors. Now we will understand about ligand gated ion channels. Okay, so ligand gated ion channels are kind of ion channels. First type of neurotransmitter means the neurotransmitter which act first, they actually act through this kind of channels. Okay, so fast neurotransmitters actually act through this kind of channels, and the receptors which are present in the plasma membrane or in the cell membrane are ion selective in nature. Means the receptor which are present in your plasma membrane or cell membrane, they are always ion selective in nature. Okay, so after binding of agonist to that receptor, ion channels will open and the alteration in membrane potential will occur, or in the changes in the intracellular ion concentration will occur. As a result, cellular activity changes. Okay, so we can write like that after binding of agonist with the receptor, it actually causes the alteration in ion channel or opening of ion channel. As a result, membrane potential changes and intracellular ion concentration changes. Ion concentration occurs. Okay, and thus the cellular activity started. Now we will see what is the example. Actually, GABA A type of receptor act through this channel, this type of ligand gated ion channels, and the ion channels which is responsible, that is chlorine channel, Cl minus channel or chlorine channel. Okay. Now the second type is that is enzyme linked receptor. Okay. So this kind of receptors actually consist of two type of domains. Okay, or two parts. First one is extracellular domain and second one is intracellular domain. It means first domain is present outer part of the cell and second part is present in the inner part of the cell. Okay, so first is extracellular domain where agonist binds. Okay, and the second one is intracellular domain where effector will bind. Okay. Now we will assume that agonist which is binding is insulin and effector which is binding, that is tyrosine kinase. Tyrosine kinase enzyme. Okay. Now we will understand that how this insulin like agonist and effector like tyrosine kinase act. Okay. When the insulin or agonist binds with the tyrosine kinase receptor, which is primarily present as a two single moiety or two single part or individual part, aggregates to form a dimer. Okay, previously they present as a two single moiety or two single part, and after binding with the agonist they will form a dimer. Okay, and as a result subsequent autophosphorylation occur. Then this activated autophosphorylated dimer binds with a relay protein and activates the protein. Okay, after activation of the relay protein, that protein triggers the cellular response through either production of a second messenger or turning on gene expression. So when insulin binds with the tyrosine kinase receptor, which is present actually two individual domain or single domain, or you can like write part, forms a dimer. Okay, aggregates and forms a dimer. After formation of dimer, autophosphorylation will occur, and this autophosphorylated dimer binds with a relay protein. Activates and binds with the relay protein, and this relay protein actually trigger the cellular response, either production of a second messenger or turning on gene expression. Okay, now we will understand it through a picture. This is the cell membrane or your plasma membrane. Agonist binding site, this is a single individual part, and these two parts are tyrosine kinase receptors, and these are the tyrosine residues. Okay, tyrosine kinase receptor. Now, after binding of agonist like insulin, it actually forms a dimer. Okay, this is your plasma membrane. This is your tyrosine residue, and phosphorylation occur. It means phosphate molecules bind to that tyrosine residue. Now, after binding, the relay protein which are present in the intracellular part, these are the relay proteins. This relay protein will bind with that orthophosphorylated dimer moiety. Okay, and as a result, this protein will trigger the cellular response. Clear to you? Now we will understand the another classification, that is your G protein coupled receptor or linked receptor. Okay, so neurotransmitter which are slow in nature, they actually act through this channel. It means slow type of neurotransmitter act through this channel. Okay, now how they act? Actually, first, the extracellular ligand is selectively detected by the cell surface receptor, and the receptor in turn triggers the activation of a G protein which is located on the cytoplasmic phase of the cell membrane. Clear? The activated G protein then changes the activity of an effector element, usually an enzyme or ion channel, and this element then changes the concentration of the intracellular second messenger. We will understand step by step. So first, what I have said, the extracellular ligand is detected by a cell surface receptor. Okay. So extracellular ligand is detected by cell surface receptor. Then this cell surface receptor triggers the activation of a G protein. Okay. Then activation of G protein occurs, which is located in the cytoplasmic phase of your cell membrane. It means your cytoplasm, inner part of your cell. After activation of G protein, it actually activates the effector element or changes in the activity of an effector element, which can be an enzyme or it can be an ion channel. So changes in the activity of effector element. After activation of the effector element, this effector element then changes the concentration of intracellular second messenger or second messengers which are present inner part of your cell. Clear to you? That is the pathway of G protein coupled receptor. Okay, signal transduction pathway. Now we will see what are the examples. First one is that is GS type of. Okay, so this GS type of G protein actually have a stimulatory effect. Okay, and it acts actually by increasing the level of cyclic AMP. Okay, and the receptor which is act through is it is beta type of receptor. And the second one is GI type. It actually inhibitory in nature, and the receptors which act through it are alpha and M2, which is actually decreases the cyclic AMP level. Clear to you? Another one or third one is GQ. GQ also stimulatory in nature, and Q actually represents a type. Okay, and the receptors which act through is alpha, that is alpha M1, M3, which actually liberates DAG and IP3, or diacylglycerol and inositol triphosphate. Now we will see what are the effectors which are regulated by G protein coupled receptor. Okay, so first effector is adenylyl cyclase, which actually forms the second messenger, that is cyclic AMP, which activates protein kinase A with phosphorylation process. And the second one is phospholipase C, which actually liberates second messengers like DAG, IP3, or diacylglycerol and inositol triphosphate. Okay, and DAG activates protein kinase C, and IP3 stimulates Ca2 plus release from sarcoplasmic reticulum. Okay, now we will understand everything step by step. Adenylyl cyclase actually forms second messenger, that is cyclic AMP, then which activates this cyclic AMP activates protein kinase A, okay, or pKa, and phosphorylation occurs. Okay, and phospholipase C liberates second messengers like DAG and IP3. And this DAG actually activates protein kinase C type, and IP3 stimulates the calcium release from the sarcoplasmic reticulum. Another three type of effector is channels, and the receptor which act through it is M2 receptor, which actually coupled to GI type of protein and causes calcium outflow. Okay, sorry, potassium outflow. And M2 type actually act through GI type of G protein coupled receptor, and calcium outflow will leads to hyperpolarization of your cell membrane. Another type, last one, is nuclear receptors, where nuclear chromatin or DNA is involved. Okay, so when the steroid hormones, estrogen, thyroid hormones, vitamin D, these kind of molecules enter the target cell, it will combine with the intracellular proteins, receptor proteins. Okay, then the intracellular receptor protein, which is associated with the nuclear chromatin, will activate or inhibit the transcription of the nearby gene. Okay, now we will understand it step by step. When steroid hormones, thyroid hormones, vitamin D, this kind of molecules enter to the target cell, they combine with the intracellular receptor protein. Receptor protein. And this receptor protein which is associated with the nuclear chromatin or DNA. Which will then activates or inhibits the transcription of the nearby gene. So this is the end of my lecture. If you like my video, please do like, comment, share and subscribe. Thank you for your attention.